Well, in this video we want to talk about one of the techniques with using the fluorescence phenomenon, which is known as the fluorescence resonance energy transfer, known as FRET. The idea behind that is too simple. We want to use two different fluorophore molecules. This molecule can be excited by a specific wave and then it will emit another wave. If the emission wave of the first molecule is the one which can also excite this one, we can somehow see the conformational change in the structure of the molecules or in the binding of two proteins or proteins and DNAs, and study the kinetics of the binding of two different proteins or the conformational change. But let's have a better look at what is happening. Well, as you see here, we have our first molecule and the fluorophore, which is excited and it emits the blue light. Here we have our second fluorophore, but the distance between these is more than 10 nanometer. So the energy which is emitted by this light cannot excite the fluorophore here. But when the distance between these two fluorophores is decreased, now the emission light of the first fluorophore can be used as the excitation light for the second fluorophore. And then when you get the emission light from the second molecule, you can see that they are close together. And this is the basic idea which we can use to see, for example, if two proteins are bonded together, or if the conformational change in the proteins, DNAs, or other compounds can happen.